All right, today we're going to talk about mitosis and the steps of mitosis. There'll be quite a few things to write down, and we really need to pay attention today. A big portion of our test is going to be what we cover today. So, mitosis. Why don't you write down 6 stages? There are 6 stages of mitosis. So cells divide in stages or steps. So maybe say 6 stages or 6 stages of mitosis. And there they are. So that's what we're going to talk about today. The first stage is called interphase. And I'd write down both bullets. So take a moment, do that. Remember, if I'm going too fast, you can pause it. Interphase, a cell is doing its normal cell activities. It's copying its DNA. It's doing everyday cell things in interphase. Most of a cell's life is going to be in interphase. So, if I was going to draw a cell in interphase, there it is, doing everyday cell things. So, the mitochondria is doing its energy thing. DNA is copying itself. We got the ER making ribosomes to make protein. All of that good stuff in interphase. Now, let's say a cell decides, hey, I need to make more of me. I need to make more of me. Maybe it's a skin cell, and you got a scrape on your elbow, and the skin cells in the area that were not damaged say, hey, we got to plug this thing. Let's divide. So it's going to go from interphase, where it's doing regular cell things, to prophase. Write it down, and then I will explain it. Prophase. Centrioles move to opposite ends of the cell. Chromosomes are fully visible. So if I was going to draw that, we still have the nucleus. Now you can actually see the chromosomes. So those little X's are chromosomes. There's also these things called centrioles that kind of go at the, they call these the poles of the cell. Just like the north and south pole is opposite ends of the globe. Okay, same thing here with the cell in prophase. Probably wouldn't hurt to sketch these in your notes. So we have the centrioles, the poles, these little points going here, and then we have the chromosomes in the center. You can see them. Normally you can't see them. You can just kind of tell some things are there. Here you can actually see the X's, the chromosomes. That is prophase. So we have interphase, then we have prophase. Then we have metaphase. Go ahead and write this. The chromosomes are going to line up in the middle of the cell. Spindle fibers attached to chromosomes at the centromere. Write this down, I will explain it. So let's draw a cell in metaphase. All right, so we got the poles here. Now the nucleus is kind of gone, and these chromosomes are going to line up like that. Okay, now if this was a human cell, there would be 23 of these X's, 23 pairs of chromosomes. For me and my drawing skills, it's probably best if I just do a few. Okay, now there's these little string-like things, they call them spindle fibers, and they're connected with the chromosomes to the poles of the cell. So it looks like that. These are little strings that are attached here. And they're kind of playing tug of war with the chromosomes. And this is metaphase. So they're playing tug of war with those chromosomes. They're all lined up there, and those are spindle fibers. All right. Next, we have anaphase. Spindle fibers pull chromosomes apart. They're going to continue playing tug of war, and they're going to pull those bad boys apart. So if I draw anaphase here, there's the poles. These are chromosomes that were pulled apart. So they're kind of playing tug of war, and no one really won, but what happened was they pulled them apart. Those chromosomes pulled apart. So those X's, those pairs of chromosomes, each got split. This is anaphase. Then we have telophase. New nuclear membrane forms, spindle fibers disappear, chromosomes are less visible. Write that, please. Telophase kind of looks like Siamese twin cells. Now they pulled these chromosomes to each end here. So they played tug of war, and they pulled chromosomes this side, and each side became a new cell. So it kind of looks like a Siamese twin. Okay, they have their own nucleus. They're having their own other organelles, but they're still kind of stuck together. That would be telophase. And then cytokinesis. This is the final phase here. The division of cytoplasm and distributing of organelles to each new cell. So cytokinesis, then it actually splits into two brand new cells. If I'm going too fast, please pause. So if we were going to draw that, we would have two small cells. These are exact copies of each other. They're an exact clone. So we have two identical cells. Now, these cells are going to grow and get bigger and bigger, and then they're both going to be in interphase. And now we have 2 cells in interphase. And then let's say they wanted to split, so those 2 cells would go copy themselves in interphase, go pro, meta, ana, tele, cyto, and then we'd have 4. And then if they wanted to do it again, we could go through and there'd be 8 and 16 and so on. And that's how you heal wounds, okay? That's how if you get a scrape on your skin, that's how that fixes itself. Your skin cells multiply like this. This whole thing is called mitosis. Okay, it might be something like with your bones. Maybe you're growing, okay? Your bone cells need to make more. They're going to do this. Maybe you just have some dead cells that your body's trying to replace. They're going to go through mitosis, okay? So you must know and understand these steps in cell division. Also know that this original copy, these are the exact same, exactly the same. Okay, they are clones, they are twins. 2 identical cells. That is all for today.